So, pag sinabing unidirectional, in one direction lang, hindi siya bidirectional. So, hindi naman pwedeng mag-donate ng fertility factor si recipient. Kaya nga recipient, di ba? So, after replication, the double-stranded DNA is present in both cells. So, in this case, the recipient also contains F factor. Anong meron sa F factor? Fertility factor. So, that is your conjugation through sex pilus. In grape clusters, okay, pag staph, clusters, kapag strep, in chains. Kapag Diplococcus, Vitus, okay? Kapag Sarcina, ito yung 3D geometrical form. So, para siyang apat na coccus. So, dalawa dito, dalawa. Then sa likod, may dalawa pa. So, parang 3D ang orientation niya. And may si monococcus, so they come singly. Okay, Bacillus anthracis. So, Bacillus anthracis is streptobacilli. So, they come in chains, rods in chains. Spore central, with central spore. Okay? Contributes to the negative charge of the bacterial surface, lipopolysaccharide. Naulit lang to sa quiz. Kinapi ko lang. Okay. Bacteria or bacterium that lack cell walls is or are aorta. Lahat. Mycoplasma and ureaplasma do not contain cell wall. Ano ang counterpart ng cell wall sa mga walang cell walls? Your sterol. So, aorta. So, site of bacterial respiration and photosynthesis, that will be your plasma membrane. Plasma membrane. So, definition lang to. A bacterial structure that gives microbe resistance to tetracyclines and chloramphenicol. So, these are your plasmids. May dalawang plasmid, di ba? Small plasmid and large plasmid. Small plasmids, yan, gives resistance to tetracyclines and chloramphenicol. Your large plasmid, they give resistance to beta-lactams. Ba? Large plasmid production kasi yan ng beta-lactamases. Kung may beta-lactamase, i-cleave niya si beta-lactam, rendering your antibiotic ineffective. Okay. Okay, lipopolysaccharide, okay, sabi nga natin, contributes to the negative charge of the bacterial surface. Yung capsule, evasion of phagocytosis or the human host or the host defense. Okay, polyphosphate granules. So, board exam question to, recall. So, MTB, match granules. Kapag bipolar bodies, yan si Yersinia pestis. Kapag Babes Ernst naman, kay Clostridium diphtherium. Okay? C. diphtherium. Endospores aid in the survival of bacteria against external conditions. Clostridium tetani has terminal spore. So, central spore, si Bacillus anthracis. Subterminal spore, si Clostridium tetani. Subterminal spore, botulinum. Okay. And, si tetani is subterminal. So, i-remind nyo ako, no? Subterminal tetani. Okay, i-remind nyo na lang ako mamaya. I-chat nyo sa GC. Okay, this serves as virulence factor of the bacteria for it to protect the bacteria from the attack of human immune system. So, both glycocalyx and capsule are the same. So, your glycocalyx appears as the capsule. I-remind nyo ako sa terminal spore, or i-recheck ko na lang. Pero I think C. tetani and C. botulinum is subterminal. Okay. Single flagellum on both sides, amphitricus. When we say monotricus, one flagella on one side. Pag cephalotricus, a tuft, or group of flagella on both sides, cephalo. Pag peritricus, around. Pag lophotricus naman, a tuft on one side. Okay. Pseudomonas aeruginosa with a tuft of flagella on one side is termed lophotricus. So, kasasabi lang, the Neisseria gonorrhoeae surrounded with fimbriae is termed peritricus. So, what makes the second statement wrong? Neisseria gonorrhoeae, hindi flagella yung bumabalot, but fimbriae or your cilias. Okay, utilizes formic hydrogenolase that converts formic acid into an equal amount of hydrogen and carbon dioxide. Your mixed acid fermentation. Pag sinabi natin butane, diol fermentation, di ba? From pyruvate to acetoin. Okay, yung mga bacteria, the next question are Enterobacter, Serratia, Erwinia, Bacillus, Yan, they undergo butanediol fermentation. How about yung homolactic fermentation? From pyruvate to lactate or lactic acid. Pag butyric naman, pyruvate to butyrate. Oh, madali lang. Pag sinabing alcoholic fermentation, syempre sugar to ethanol. Okay, anaerobic. Okay, aerobic bacteria require oxygen and grow well in room with air. So blank oxygen, okay, 15 to 21%, and 21% carbon dioxide is needed. So the family Enterobacteriaceae are examples of facultative anaerobes. So ano ba yung mga aerotolerance natin? No? Number 1, si Cutibacterium, yung mga acne natin, aerotolerant. Yung si obligate, sila Clostridium, Bacteroides, ayan. Microaerophils, sila Campylobacter, Treponema, and aerobes, Bordetella, Brucella, MTB, yung Pay, Pseudomonas aeruginosa. Okay, trust the process guys, magiging familiar din kayo sa mga yun. Okay. Cutibacterium acne can survive in the presence of oxygen but cannot perform metabolic processes unless situated in an anaerobic environment. So this bacterium is known as mentioned kanina, aerotolerant anaerobes. So obligate and facultative anaerobe contains protective enzymes that counter the toxic effects of oxygen. The enzymes are superoxide dismutase and catalase. Counter the toxic effects of oxygen. Yung beta-lactamases nyo, beta-lactam drugs mga yan. So, i-cancel nyo na yan. Parang given na ito eh. Beta-lactamase, beta-lactamase, beta-lactamase. Diba? So, superoxide dismutase and catalase. All are capnophilic bacteria except Bordetella pertussis. Ano ba yung mga capnophils natin? Yan. Si H. influenzae, Neisseria gonorrhoeae, Streptococcus pneumoniae. Okay. They use the reduced and preformed organic molecules from other bacteria, yung heterotrophs. Pag organotropes, by definition, so these require organic substance for growth. Yung phototropes naman, o photosynthesis, uses light as energy source. Pag lithotropes, reduced inorganic naman. So inorganic molecule for their biosynthesis. And we say autotrophs, so carbon dioxide as the only source of carbon. So next, Listeria monocytogenes, Yersinia enterolytica, Legionella are examples of psychrophilic bacteria. Ano ba pag psychrophilic? Mahilig sa malamig. At yung Legionella natin, ito yung bacteria found on aircon, sa mga aircon natin. Halophils are Staph aureus, Listeria monocytogenes, and they require and grow in increased concentration of salt. Okay, or your NaCl. So, alam mo nyo na, nasa MSA, monitor, yes, may magtatanong ata.
May nagpapaksa po, sir. Okay.
Okay. So na tayo. Pag halophils, di ba we have your culture agar, MSA, mannitol salt agar. So, alam niyo na si SRUs and L-monocytogenes, mag-grow sila sa MSA. Kasi they are halophils. Okay. Fastidious bacteria are unable to grow due to their incapability to synthesize organic molecules and they are not easy to cultivate. So, fastidious, mabilis lang yung buhay nila, lifespan. Yan na lang para tandaan, para tandaan natin. So, cell divisions equal cell death, plot 2, di ba? Lag phase, kapag lag phase, di ba yung paangat pa lang yan? So, strictly, may mga growth naman yan. Kaso, by definition, no cell division pa lang. Sa lag phase and exponential, the same lang yan. Decline phase or the dead phase. Okay, utilizes polycarbonate membrane filter or millipore filter. Membrane filter, millipore filter, membrane filter. Okay, all of the following are motile except Pasturella. So, Providencia, Pseudomonas, Proteus, E. coli, they are motile. All of the following are non-motile except Clostridium tetani. So, yung mga iba dyang choices, they are, syempre, motile or motile. So, this bacterium or bacteria can evade phagocytosis. So, lahat yan. Ano ba yung meron sa kanila? Bakit nila na-evade yung human host defense? They are capsulated. So, Bacillus anthracis, Streptococcus pneumoniae, Haemophilus influenzae, they have capsules. The laboratory can make accurate and useful determination only if a specimen has been collected and recorded properly. And the results, okay, limited by the quality of the specimen and its condition, both are correct. Which of the following statement is correct using cotton swab? So use cotton swabs for nares and throat specimens. Yan. Common naman yan, COVID. Diba? Nagsaswab sa ilong or dito. So, swab specimens generate greater quality. Nope. Mas maganda pa rin ang needle aspirates. Okay? Gumagamit ba ng cotton swab sa nasal gonorrhea? Can be, but hindi siya recommended. So, cotton swabs provide moisture. So, hindi. Kasi dry ang cotton swab. May mga fatty acids that can be toxic to bacterial survival. Okay. Changes in oxygen, pH, temperature inhibit growth of other bacteria. At the same time, promotes the growth of other bacteria. Both are correct. Can be transported within 24 hours at 40 degrees Celsius. So your clean, voided midstream urine. So yung gastric aspirate, corneal scraping, prostatic samples, and body fluids, transported yan agad-agad. Okay? At room temperature itong mga to. Clostridium difficile toxin, preservation. So, iota can be refrigerated, stored at -70, collected without preservative. Ang naalala ko ata lumabas sa LMS nyo is stored at -700, parang ganon. Sa LMS kasi bago namin ipasok sa program or sa app na LMS, naka-plain text siya. So, yung degree Celsius binasa niya as 0. Pero, ang na-accept ko pa rin sagot is aorta. Since Clostridium difficile toxin is collected without preservatives and can be refrigerated. Okay. Hindi naman pwedeng nota. SPS except, so neutralizes bacteriostatic effect of plasma. Dapat neutralizes bactericidal effect of plasma. Okay. You receive a serum specimen for serological studies. You should store the specimen at RF temperature. Hair, nail, or skin scraping are stored at indefinitely. Okay? At room temp. So, hindi mo na kailangan i-store. Pag tinatamad ka, pwede mong hindi mo muna gawin, pero dapat gawin mo na para hindi ka matambakan. So, indefinitely, ibig sabihin, as time permits. Basta hindi pa nawawala, pwede pa yan. And stored at room temperature. You only receive one swab at bacteriology section in a good condition. You should culture the specimen, then prepare smear from the plate after incubation. Make a smear first, hindi pwede. Hindi mo pwedeng gamitin yung pinagawa mong smear tapos i-streak mo sa culture plate. Hindi pwede yun, prone to contamination. So make a smear first, so mali. Culture the specimen first, then prepare a smear. So from the same swab. So hindi pwede. Ideally, may dalawa dapat kasing sinasubmit. Pero nag-submit sila sa'yo ng isa in a good condition. So, syempre, ang gagawin mo doon, kakausapin mo na next time, 2 swabs. Pero hindi mo naman na dapat i-reject yung specimen. Kasi pwede mo na yan i-culture. Then, pag na-culture mo na, nakatubo na yung growth, kumuha ka ng colony, yun na ang gawin mong smear. Okay? Ganun lang yun. So, reject the specimen. You don't need to reject. So, Bartlett's criteria, okay, ganyan. Less than 10 epithelial cells, greater than 25 pus cells. Paano pag mas marami epithelial cells? So, hindi na yan sputum. So, that saliva na yan. So, repeat collection. So, required volume for CSF, 10 ml. So, best specimen for infants in collecting GI tract. So, best is, lahat pwede, pero ang best, gastric aspirate. May mga infants kasi, na kapag aerosol-induced, may mga malilikot na bata. So, baka hapon na, di na pa nakakolektahan kahit induced na yung kanya. Yung sputum niya. So, simultaneously produce sputum. Alam niyo naman, nung bata tayo, nilulunok natin yung prokak natin. And gastric biopsy, this is invasive kasi ito eh. So, hanggat maaari, wag muna. Kung kaya pa naman ng gastric aspirate, aerosol-induced sa mga nakikiko-operate na bata. Yan. Pero best specimen would be gastric aspirate. Okay. Kasi they tend to swallow their sputum. Okay, pure culture of E. coli greater than 105 CFU per ml. Yung mga sumunod na choices dyan, bubuo ko lang. So, negative staining except bacterial structure and morphology. What makes this wrong? Hindi mo makikita ang structures. Okay? Hindi mo makikita ang mga structures. Hindi mo makikita si DNA, hindi mo makikita si plasmid sa bacteria. Kasi ang ini-stain ng negative staining, instead na yung mismong bacteria, yung background. So, yung morphology, yung shape ng bacteria. Kaya nga ginagamit ito sa mga treponemes, yung mga spirilla. Okay? Yung mga pa ganon-ganon na bacteria natin. Mga spiral shaped. Okay. So, negative staining, pwede rin siya for gas vacuole. Ginagamit siya for gas vacuole. Example of dyes would be India ink or nitrocene. Okay, the differential process in gram staining is the colorizing step. So, cell walls with teichoic acid do not retain the primary stain after the colorization, which is wrong kasi ang cell walls with lipopolysaccharide retain the primary stain. Yung cell walls with the lipopolysaccharide is a gram negative. Gram negative na to. Kung may lipopolysaccharide, gram negative. Ano ba yung mga gram negative? Hindi nila nare-retain yung primary stain. Ano bang kulay ng gram negative? Red, di ba? Anong kulay ng counter stain? Red. So they are, they do not retain the primary stain. So with teichoic acid, so these are your gram positive, they retain the primary stain. Okay? So, gram-negative bacilli except mycobacteria. So, ano ba yung mga bacilli natin, di ba? Sabi natin, all bacilli are gram-negative except, ayun, mycobacteria, arcanobacteria, bacillus, clostridium, sino pa ba? Cordonia, Cutria, Chocamorella, yan. So, itong mga given, pang choices, gram-negative bacilli sila. Okay. So, gram-positive cocci except. So, all cocci are gram-positive except nevermore. Diba? Neisseria, Veillonella, Branhamella, or Moraxella. Okay. Differentiate Mycobacterium smegmatis from MTB, your Pappenheim method. Si Baumgarten differentiates Mycobacterium leprae from MTB. Okay? Yung aramine-rhodamine method naman, this is selective for the wall of acid-fast bacilla. Yung Kinyoun method and Zille-Nielsen, Kinyoun or hot method, Kinyoun or cold method, Zille-Nielsen or hot method, these are your acid-fast stain techniques. Tama? So 1% H2SO4 in 70% ethanol is the acid alcohol used in modified acid-fast staining, which is correct. What makes the second statement wrong? Sa hot method, hindi siya 10%. It's 3% HCl in 95% ethanol. Okay. Fontana Tribundu and Lavatitis technique stains spirochetes. Ano yung mga techniques natin sa capsule? We have Anthony's, his, Jeans, flagella naman, Leifson. Okay. Cell wall, si Jar, and metachromatic granules would be si Nasser, tama, and Albert. Tama ba? Check natin. Pero tatingin ko tama. So, yun na. Nasser and Albert techniques, tama. Metachromatic granules. Okay. All of the following for cup are true except cup is made up of 2 to 3% chocolate. Nung unang encounter na question ko to nung college, hindi ko minind itong chocolate. Akala ko may chocolate talaga. Pero hindi. Yung kaya nagbe-brown yun, lysed RBC. Kasi ang cup nyo. Kaya siya nag-brown. Okay, tissue culture medium for human epithelial cells of larynx carcinoma, your HEP2 cell line. HeLa 229 from human cervix. McCoy, fibroblasts. Diba? Vero cell line, kidney cells of the monkeys sa Africa. Diba? And A549, human lung carcinoma. Kwanto, lumabas daw to sa board exam, past years, long years ago. Pero sa amin, wala. So, culture media that is used to propagate the growth of certain groups of bacteria. Okay, your enrichment medium. Okay, routinely used for the recovery of Haemophilus species. Routinely used, So, ito yung parang first line. Ito muna yung gagamitin mo, si cap. Okay, pero si Haemophilus influenzae, nag-grow din kasi siya sa iba pang cap. Specifically, sa iba pang plate, sa iba pang agar. So, cap, example is cap, specifically yung may horse blood. Okay? So, pwede rin yung Haemophilus sa fetal bovine serum and rabbit blood agar. Pero dapat sa rabbit blood agar, meron kang yeast extract. Tama? Nasa kwanto, table. Alkaline peptone water adjusted to 8.5. Okay. Promotes growth of Vibrio species. Okay. Used as both an enrichment and selective medium that contains sodium citrate and sodium desoxycholate that inhibits gram-positive bacteria. So, your gram-negative broth, ini-inhibit niya daw yung gram-positive. So, para saan siya? Sa gram-negative. So, MAC agar differentiates lactose fermenters, yellow, from non-lactose fermenters, pink. So, baliktad, no? Pag lactose fermenters, pink. Kapag fermenter, pink. So, neutral red as the pH indicator. This detects the fermentation of sugar and eventually the production of acid. So, Columbia colistin nalidiksik agar. Guys, i-memorize nyo din yung ano, no? Kapag CNA, dapat alam yung ibig sabihin ng CNA is Columbia colistin nalidiksik agar. Sa boards, ang mga choices hindi sa in-words, pero naka-CNA, BAP, EMD, MAC, MHA, ganun. So, pero may mababait na mga examiner na i-spell out nila yan. But to be safe, you should be familiarized then. So, gram-positive bacteria, specifically mga gram-positive cocci, your CNAs. You are to make selective media for gram-negative bacteria. All of the following reagents are to be included except sodium azide. Sodium azide, ini-inhibit niya yung gram-neg. So, gagawa ka ng plate for gram-neg, huwag mo ilagay si sodium azide kasi si sodium azide, ini-inhibit niya ang growth ng gram-neg. Si crystal violet, carbolfuchsin, balsol, gentian, they are all inhibitors of gram-positive. Okay, protein-rich media, which is composed of whole eggs, malachite green, LJ medium, matic, whole eggs. Okay, anong kulay ng LJ medium? Greenish siya, di ba? Malachite green. Okay, growth for mycobacteria. Okay, no questions, LJ medium. For urine specimen, ang calibrated loop at 0.01 ml. So, incubation of anaerobes, pag 5 to 10%, yan, anaerobe siya. 5 to 10% H2, 8 to 9% N2. How about yung 21% oxygen plus 0.03% carbon dioxide? Kanino? Incubation for your aerobic naman. Diba? Yung 5 to 10% carbon dioxide plus 15% oxygen, yan yung capnophils. Okay, capnophils, mga capnophils. Yung following choices, uubo lang yan. Incubation condition for microaerophils, 5 to 10% oxygen, and 8 to 10% carbon dioxide. Okay, watch out lang, pag 15% oxygen, 5 to 10% carbon dioxide, kahit kanino ulit, capnophils. Kapag 5 to 10% oxygen, okay, and 8 to 10% carbon dioxide, microaerophils. Okay. In an anaerobic chamber, blank is responsible for removal of residual oxygen, your palladium pellets, diba? Or your catalyst. Ito yung catalyst natin, palladium pellets. Ano bang action ng silica gel? Ina-absorb niya yung water. Okay? Kapag yung hydrogen nag-combine siya sa oxygen in the presence of your palladium pellets, i-absorb niya. Nitrogen gas, fillers lang to sa loob ng chamber. Bisazoline and methylene blue, sila yung color indicator. Pseudomonas aeruginosa has dendritic pattern, correct? Sprotius, concentric rings, correct. Cultural characteristic, elevation, except entire. Si entire kasi is for margin. Okay. Biosafety cabinet wherein 70% of air is recirculated, 30% exhausted. 2A1. Si 2B, sila yung mga toxic, carcinogens, radioisotope, yun sila yung, ito yung mga cabinet ito yung cabinet na gagamitin. Plus 2B. Kapag 2B1, 30%, baliktad lang. So, 30% naman ang non-recirculate sa 2B1 at 70% naman ang na-exhaust. Binaliktad lang. Ay, so BSC for chemicals, radioisotopes, 2B. BSC wherein 30% of air is circulated, 2B1. So BSL3 agents exact, HBV. So HBV is level 2. So sino pa ba sa level 2? HIV, Bacillus anthracis, Yersinia pestis. Yeah. Category A agents sa bioterrorism, smallpox. So si rickettsia, category B yan. Sino pa sa category B? Coxiella burnetii, Burkholderia pseudomallei. Sino pa ba? Yung St. Louis virus, encephalitis virus. Okay, culture media in the lab are checked based on their performance and sterility. Records are kept for 2 years. So autoclave, media, liquids, utensils, glass pipettes at 121 degrees Celsius, 15 psi for 15 minutes. So pasteurizing milk at high temperature will give an advantage, which is milk can be stored in temperature for 2 months without affecting its flavor or ultra high temperature. Okay, phenol coefficient greater than 1. So disinfection is more effective than phenol. Kapag phenol coefficient less than 1, ibig sabihin, phenol is more effective than disinfectant. Kapag both phenol and disinfectant has the same efficiency, PC is equal to 1. CRO, your ceftriaxone, your E, erythromycin. All of the following are beta-lactamase-resistant antibiotics except amoxicillin. So, beta-lactamase resistant. Nalala niyo pa yung mnemonics, si Dominic or Dominac. So, D for diclosoxalin, O for oxacillin, M for methicillin, N for nafcillin, and C for cloxacillin. So, protein synthesis inhibitors binds to 30S and 50S ribosomal subunits of bacteria. 40S and 60S ribosomal unit naman natin yon ng mga eukaryotic cells. 30, yung mga following choices, buo-buo ko lang yan. So, this antibiotic may cause temporary or permanent depression. So, your chloramphenicol. Nucleic acid inhibitors except penicillin. So, penicillin, kung hindi mo na-review yung nucleic acid inhibitors, ang penicillin naman, parang siya na yung major example ng cell wall inhibitors. So, kakancel mo na siya agad. Okay? All of the following are transmitted through sexual contact except Neisseria meningitidis. So, gonorrhea, syphilis, T. pallidum, C. trachomatis, and HIV are your STIs, or sexually transmitted infections. So, Viridans streptococci are normal microbiota of mouth and oral cavity. So, in immunocompromised patient, even normal microbiota can cause infection. This is termed as true pathogen. So, only the first statement is correct. So, virulent streptococci, normal microbiota ng mouth and oral cavity. Pero, kapag immunocompromised yung tao, itong mga normal microbiota can still infect na yung tao. Kasi nga, immunocompromised, humina yung immune system. So, yeah, hindi sila, so they took the opportunity na humina yung immune system mo. Therefore, hindi sila true pathogens. They are opportunistic pathogens. Tama? Kasi hindi naman sila pathogenic eh. Naging pathogenic lang kasi humina yung immune system mo. Kinuha nila yung opportunista. The opportunistic pathogens. Okay, the toxin represents gas gangrene, tetanus, botulism, diphtheria, and scarlet fever. Exotoxin. Ano naman yung mga disease represented by endotoxin? Typhoid fever, UTI, meningococcal meningitis. Tama? So, antigenic shift, both major genetic change responsible for epidemics. How about sa antigenic drift? Sila ay minor, tama ba? Minor genetic change that is responsible for both epidemics and pandemics. So, DNA elements that encode transposition and excision functions and which are also able to carry antibiotic resistance gene among plasmids and chromosomes, transposons by definition. So results of a biochemical makeup of wild-type organism depends on hydrophobic, hydrophilic, so intrinsic resistance. Definitions lang to. So dental plaque and periodontal disease can be thought of a continuum of what type of physiological process. Ano lang ba yung physiological processes na nabanggit natin? Process na nabanggit natin sa discussion? Biofilm production or biofilm formation. And streptococcus pneumoniae can be part of normal flora of 5 to 40% of people. At what anatomic site? Nasopharynx. Strep. Dito. Hindi naman pwede sa vagina, urethra, colon. Kaya nga, streptococcus. Alam niyo yung strepsils? Para alam natin na ano yung bacteria dito, streptococcus. Ano yung candy na ano. Ito may strepsils ako dito. Strepsils for streptococcal infection. Nasopharynx. Saan ba ang pharynx dito? Okay, resident flora is commonly found in urethra, liver, kidney, salivary gland, gallbladder. Hindi, mali. So certain microorganisms are never considered to be members of normal flora. So they are always considered to be pathogens. Which of the following fits in the category? So ano yung pathogenic dito? Mycobacterium. MTB. Kapag may MTB ka, kahit isa lang nga na makikita sa sputum, kahit isa lang, naku, i-ano na yan, i-isolate na kasi, isolate sa room kasi pwede pang kumalat. Okay, streptococcus pneumoniae, sabi nga natin dito, yung Escherichia coli, meron yan sa guts natin, sa intestines, sa streptococcus, Staphylococcus aureus, meron yan sa skin, Neisseria meningitidis, normal flora. So we don't talk about Bruno, hindi niya pa ba napanood to? Yaman niyo kung bakit may mga nakamali pa dito. We don't talk about abuela. May sumagot. We don't talk about anymore. Di ba? Yung kanta naman, we don't talk anymore. May iota din, pero it's Bruno. Yung hindi sa lahat ng bagay umiikot yung buhay natin sa sa studies. So always find time din naman para makarelax or para makabawas sa stress. Ako, I'm coping up, playing mobile games para hindi laging school. Kaya ano mga coping mechanisms niya para hindi ka masyadong ma-stress? Tulog. Mas better, tulog. So, ayun. Pinopromote ko na panoorin yung Encanto pag di niya pa napanood. Maganda. Yan. May mga mali pa rin, naalala ko talaga, makuha to lahat. Anyways, may 2 points na ata, plus yung Clostridium tetani kanina. Matching type, ina-expect ko din na 10/10 most. Pero bakit kaya sinabi ko naman na may 10 to 15 points? Buti na lang hindi nga road 15. So yeah, BCYE, Legionella species, BSE, selective for Salmonella, footpad of mice, Mycobacterium leprae, Cetrimide, Pseudomonas, MAC with sorbitol, 015707, MSA, okay, sabi natin kanina, Staph au and Yersinia are halophiles. So, mahilig sila sa salt. So, MSA, Staph, Regan-Low, Bordetella, Thier-Martin, or TMA, Neisseria gonorrhoeae. Actually, meron pang modified Thier-Martin So, for Neisseria gonorrhoeae, Tinsdale, Cyanobacterium, XLD for Salmonella and Shigella. Pero marami namang hindi ko lang na-check kung marami, pero may mga naka-perfect naman dito. So, it's a good start na alam natin yung mga culture plates ng mga iba't ibang bacteria. Okay, true or false? Semi-solid medium is used for bacterial motility observation, detection of indole and sulfide production. So, true. mgRNA1 and magnesium RNA crystal violet iodine complex is easier to remove than CVI complex or crystal violet iodine complex. False, kasi kung meron si magnesium RNA, it is harder to remove. So, gram staining is not recommended on specimen from vagina, cervix, anal clips for Neisseria gonorrhoeae identification. True, boric acid maintains the appropriate colony counts for your specimen at room temperature. 24 hours, true. Facultative anaerobes, most clinically significant, true. Prokaryotes also have membrane-bound organelles. So prokaryotes are your bacteria. Wala silang membrane-bound. Okay. Swabs can be inoculated directly to culture media. True. So male and young employees. True. There is resident flora in synovial fluid. False. Sterile ang mga yan. Synovial fluid, serous fluid, cerebrospinal fluid. Sterile sila. Endotoxin, they require bacterial death to be released into circulation. So that ends yung ratio natin. So, hi, kamusta tayo? Actually, nung chinek ko yung, nung hindi ko pa na-complete yung grade nyo, pero nung chinecheck ko yung scores nyo, doon pa lang nag-sink in sa akin na 120 items pala yung exam nyo. Akala ko 100 items. Kaya sabi ko, pag 60 to 65, sige, mag-a-adjust tayo. Ewan ko, ba't last week ba't 100 items lang nasa isip ko. Pero recently ko lang nalaman na, 120 items pala itong mga to. So, yeah. And gusto kong i-share yung mga response nyo. So, nice! I like the confidence. So, malay nyo, dapat nga dito kayong lahat eh. Dapat wala na sana dito. Dito lang. Malay nyo, sino ba to? Bigyan natin ng magandang grade. Ba't dito? Ba't naman? Well, pag i-assess mo naman sarili mo, alam mo naman kung saan ka na talaga dito. So, ang average nyo nasa 80 to 85. Well, who knows? Kung saan talaga kayo. Yeah. So, gusto ko sanang kumustahin muna yung exam. So, saan dito ba si Miss Navarro? Okay ba yung exam? Miss Navarro? Dito siya. Ay, kamusta yung exam? Ikaw kasi andito sa nakikita ko. Meron ba siya? Ayan, meron siya naman. So, how's your exam?
Medyo mahirap po.
Medyo mahirap. Ako din, nung UA, urinalysis pa yun eh. Sabi ni Sir namin, ni Sir Anthony, ang dali ng exam nyo, ina-expect kong mapaperfect nyo. So, nung pagdating din sa amin, ba't ganun? Sabi ni Sir, madali lang. Scam naman si Sir. Pero, siguro, let's say, kami kasi, naka-review na kami ng board exam. So, parang naging clear na lahat. Pero, yeah, sabi ko nga, trust the process. Magiging familiar din kayo sa mga to. No. Kaya nga, mas magandang mahirapan na tayo sa kare-review or kami memorize, kakakape. Mahilig ba kayo sa kape? Ako kasi hindi. Ayoko nagkakape. Pero sanay naman ako sa puyat. Nagkakape lang ako pag kakailangan ko na talaga. Parang okay na rin para kasi may mga, yung nasobra na sa kape, hindi na pwedeng isang kape. Nakakadalawa na sila para lang magising. So, pag nagkape ako, hanggang umaga gising ako. Maghapon pa yun. So, magandang advantage. So, yeah. Sana Section B kayo, right? Sana wala na yung mindset natin na pag Section B, iisipin nyo na mas matalino pag Section A. Sana wala na yun. Kasi kami yung 4th batch at ang sakit daw ng mga medtech nung first batch, second batch, third batch, lagi silang nag-aaway, yung mga Section A, Section B. Sa amin naman, wala, neutral lang ata, pero meron pa rin yung iba. No? So, hindi porke magkaibang sections, porke Section A, mas matalino. So, sana wala na yung thinking na gano'n. Pwede namang, andito, kasi ako Section B ako noon. So, pero kita nyo naman, okay naman. So, wala naman akong inulit na subject. Si Sir Justin, Sir Argyle, Sir Giles, Section A sila nun. So, yeah, I think hindi kaso ang section. Kini-clear ko lang baka kasi meron pang nagkakainitan pa rin ba yung sections nyo? Hindi naman siguro, no?
No
Lalo
po,
na,
sir.
Oo, better yun. Mas maganda yun. Kasi noon daw, to the point na parinigan sa chance, face to face. Yung iba, lumipat, yung iba. So, may gusto kong i-clarify, gustong itanong. Kasi ang next topic natin is i-discuss na natin siya, your AST, antimicrobial susceptibility. So before sana tayo itikin topic na yun, mag 10-minute break muna tayo. No? You can have your lunch, early lunch. So, meron. So, i-remind ko lang yes, nasabi ko na mag-adjust tayo, pero sana wag na tayong umasa lagi sa adjustments. Kasi yung highest nyo is 94. So, mahirapang, may nakakuha kasi ng 40 below. So, I don't know, bawi na lang sa second sem. So, marami pang chances. Pag sinabi ko marami pang chances, may next school year pa. Pwede nyo i-retake another chance. Pero pwede namang ayusin na this second term and finals para hindi na ulitin. Okay? So, hindi naman ako strict, pero pag deserve mong makapasa, ipapasa natin. Pero pag online kasi, hindi mo makikita kung attentive yung student eh. So, hindi ko rin ma-assess kung kailangan mong ipasa or hindi, kung uma-attend ba or nakikinig naman. But thankfully, may monitoring of attendance. So, okay. So, balik tayo by 11:30. Okay, have your dinner na have your lunch para makaprepare na tayo. Okay, see you at 11:30. You can exit muna to conserve your bandwidth. So, may kainan mamaya. Kaya lang, pagpasensya niyo na yung background noise, no? Kung medyo disturbing. Kasi ako din hindi ako makafocus pag may maingay sa ano. Anyways, before pala ito, is everyone aware na we are using the same link every meeting? This one. I-click nyo lang to, 10:30, 1:30, ang klase natin. Kapag di ko kayo mamimit, I'll announce it ahead naman. No, so dito lagi. Okay? Hindi to nababago. Same lang. Okay. Ayaw na. Okay. So, welcome sa second term. Yan, and congrats. You made it. But let's see if your grade ay sumang-ayon. Anyways, 25%. Pero malaking bagay na yun. Okay, today ang pag-uusapan natin is your antimicrobial susceptibility testing. From the previous term, binigyan na natin to ng birds, especially sa staking of plates. We all know yung purpose of AST na. Okay, na-discuss natin to sa antimicrobial resistance, right? Purpose of AST, para malaman kung anong antibiotics ang dapat at hindi dapat. Okay, their zones of inhibitions. You also know that MHA is the most commonly used agar in AST. And we also have methods like your broth dilution method, agar dilution method, disc diffusion method. Ito yung disc diffusion, ito yung ginagawa sa lab. So I think we will focus more on this disc diffusion method or Kirby-Bauer test. Broth dilution method, agar dilution, okay, for the sake of information, kasi dapat malaman nyo din kung ano to. So, kasi parang yung method sa broth dilution and agar dilution will be the foundations ng ibang methods. Okay, also, you have your minimum bactericidal concentration test, time kill assay, synergy test, serum bactericidal. Honestly, hindi ko pa ito na-try, pati nung school days, dito pinatry sa amin sa tests. Pero, if you have the opportunity, which is very nice, yes, Sir Benson.
Napindot lang po, sir. Sorry po.
Okay. Kala ko may questions. Asa na tayo? Minimum or MBC test, time kill assay, synergy, serum bactericidal. Sabi ko nga, we never tried this. Pati mismo sa trabaho niyo na, hindi na ito ginagawa. Kasi ba't pa gagawin yan kung meron namang this diffusion method which is more convenient. Yeah, and better results, this diffusion method. Itong mga test na to, min MBC, time classes, synergy, zero bactericidal, we'll just run down through it, we'll discuss it a bit, and para naman may information kayo kahit papaano. Even sa review, hindi na to hindi na, yun, hindi na tinatake, should I say. And elect test, hindi nakalagay, si E-test. Meron si E-test. Okay, EST, this is used to determine whether the growth of bacterial isolate will be inherited by the desired antibiotics or develop resistance. So, saan dito ang mas better na result? Yung nasa right. Diba? Kasi yung nasa left, sabi ko nga noon, this is a very sad story. Kasi most of the antibiotics na ginagamit mo na resistant na yung bacteria. Tignan nyo, wala nang zone of inhibitions yung iba. Diba? Nakikita nyo yung arrow ko, itong mga to, wala na. Wala na, wala na. Unti. Ay, unti, ang unti, ang unti. Compared dito, okay, you can see na effective pa sila, no? Depending on the measurement. Kasi gagamit ka dyan ng vernier caliper or a ruler, okay, pwedeng ruler to measure the zone of inhibition. So, we see zone of inhibition from this point to this point. So, yan yung zone of inhibitions. Tama? Tama. So, purpose of EST. So, this one is a plate, EST plate, uninoculated. So, ganito, EST plate, MHA plate, uninoculated, yung Mueller-Hinton agar. Okay. So, this determine the capacity of bacterial isolate to cause disease. So, identify the susceptibility patterns of bacterial isolate to the antimicrobial agents and ascertain the availability of the standard susceptibility methods. So, in simple terms, pag nagpe-perform ka ng AST, ito rin yung way to control the antibiotic na magagamit mo, na i-recommend ng clinicians rather, okay, in the choice of drugs for treatment of certain infections. So, kunwari ang measurement, theoretically kunwari ang measurement nitong antibiotic na to is 14 millimeters, which is pasok siya sa range. So, kunwari ito, mga nasa 17, 10, yung mga iba niya. Ito lang yung pasok. So, ito yung gagamitin mong antibiotic. So, it can be any antibiotic like your erythromycin, clindamycin, penicillin. Okay. So, MHA or the Mueller-Hinton agar, the standard susceptibility medium 4, non-fastidious bacteria. Recommended medium 4, it has reduced concentration of the sulfonamide, trimethoprim, and tetracycline inhibitors. Note of this, sulfonamide, trimethoprim, and tetracycline inhibitors. So, this is also composed of beef infusion, nucleic acids, vitamins, casein, hydrolysate, and agar. So, ang tandaan nyo na lang, EST pinaperform para hindi basta-basta yung antibiotic na gagamitin ni patient. Okay? So, MHA, broth with 2% sodium, Mueller-Hinton broth with 2% sodium chloride is used to improve the detection of oxacillin-resistant staphylococci. Your MH broth with 5% lysed horse or sheep blood, ito yung ginagamit for testing streptococci, Neisseria meningitidis, and other fastidious organism. And we also have your terminologies. So when we say breakpoint or cutoff, this is the concentration of an antimicrobial agent that coincides with the susceptible or intermediate MIC breakpoint for a particular drug. Your MBC, MIC, and MLC. Sige nga, mag-graded recitation nga tayo. Okay, para meron agad, no? Meron tayong pambawi agad sa second sem. So anyone who can differentiate the three. Okay. Sino? Meron? Or tatawag na lang ako ulit? Tawag ako or meron? Tandaan niyo pa ba si minimum bactericidal concentration, minimum inhibitory concentration, minimum lethal concentration? Okay. Tawag ako. Kenneth Ralph Francisco. Ano ba si Kenneth? Kenneth? Wala. Andrada K. Kahit isa lang nga, Rod. Sige. Wala din. Cyrene Binuwag. Wala rin. Ba't wala sila? Buyukan. Ayun, Faith Lade Buyukan. So, andito siya. When we say MIC, MIC is, or MBC is, MLC is, ako na nga. Okay, pag sinabi nating minimum bactericidal concentration, ito yung lowest concentration ng antimicrobial agent na kailangan mo para pumatay ng bakterya. So, ito yung pinakamababang concentration ng bakterya. So, kunwari may 32, may 16. Yung 16, kaya na niyang pumatay. Gagamitin, saan doon yung MBC? Yung 16. Okay, yung MIC, lowest concentration of antimicrobial agent, that inhibits bacterial growth and does not necessarily kill. So, yun ang pagkakaiba nila. Your MLC, same lang yan sa MBC. Lowest concentration of antimicrobial agent na kailangan para pumatay ng bacteria. MBC, MLC, same. And we have your paradoxical or eagle effect. This is decreased bactericidal activity at a higher drug concentration. Yung picture dyan, okay? Ay, mali. Okay, yung picture dyan is the E. coli demonstrating eagle effect. So, ganyan yung itsura ng eagle effect. Okay, this photo is from, yan, yung source natin. When we say paradox kasi, paradoxical eagle effect, self-contradicting. Okay, ang nangyayari dito, di ba ito yung antibiotic nyo? So, you near the area of your antibiotic, mataas ang concentration. Habang lumalayo yan, pababa ng pababa yung concentration ng antibiotics. Sa paradoxical effect or the eagle effect, okay, di ba pag mas marami ang antibiotic, mas effective dapat. Pero ang nangyari sa paradoxical effect, sinelf-contradict niya. Kung saan pa yung mataas ang concentration, doon pa may growth. Kung saan pa yung mababa ang concentration, doon pa may inhibition. So, yun yung paradoxic effect. So, remember lang na pag yung drug concentration decreases at the drug concentration decreases at increasing distance from the disc. So, ibig sabihin, yung mas malapit sa disc, mas mataas yung concentration ng drug. So, in this case, yung TPZ natin, that is your tazobactam or your piperacillin. Okay, skipped wells, these are involved at higher concentrations and no growth at one or more of the lower concentrations. So, skipped wells, this may indicate improper incubation or improper concentration of antimicrobials. Kunwari ito, may growth, wala, then may growth. So, theoretically, ganun. May na-skip na well. Yun. Maintindihan naman agad. So, tolerance, the ability of organism to be inhibited by the drug, which is normally bactericidal, and when we say trailing growth, heavy bacterial growth at lower concentration that is followed by one or more wells. Okay, first method, your broth dilution method. Okay, should we... Wait lang, check ko. Ayan, may method siya. So, yeah, involves challenging the organism of interest with antimicrobial agents in broth that determines the MIC or MLC concentration. But in general, broth dilution dinedetect ang MIC para malaman mo yung minimum inhibitory concentration. Okay? Each principle, this determines the MIC or the lowest concentration of antimicrobial drug that will inhibit bacterial growth. So this is preferred method. Preferred method is a serial twofold dilution. When we say serial dilution, pababa ng pababa yung concentration. So let's take this as an example. So, kung halata dyan, ito 64 dati yung concentration ng antimicrobial mo. Then, through serial dilution, kukuha ka dito ng, let's say, 10 micrograms per ml of solution. Okay, let's say 2 ml or 1 ml, ililipat mo dito. So, mababawasan yung 64 ng magiging 32. Then, then i-mix. Then, kuha ka ulit, ilipat mo yung concentration ng drug, bababa, 16. Okay, then lipat, 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 lipat. So, bago ka maglipat-lipat, meron tong distilled water for diluting your antibiotic. Okay? So, that's your serial dilution. So, standard inoculum, 5 times 10 raised to 5 CFU per ml. So, procedure, specific amount of antibiotic is prepared in decreasing concentration in broth by serial dilution technique. So, as what we mentioned, this one, so with 64 initial concentration niya, mataas, di ba? Performing dilution. So, lipat-lipat. Bago ka maglipat, i-mix mo muna ng maayos, ha? Ayan. Okay. So, result, absence of turbidity signifies the inhibition of bacterial growth by the antibiotics being tested. So, pag may turbidity, meaning may bacterial growth. Pag walang turbidity, ibig sabihin, pag walang turbidity, no growth. So, in broth dilution, merong macro dilution and micro dilution. Pero ang ginagamit dito is yung micro dilution. So, sa broth macro dilution, so susceptibility medium, MH broth pa rin. So, broth volume greater than equal than 1 ml. Inoculum size, 5 times 10 raised to 5. CFE per ml, which is the same incubation time, 16 to 25 hours at 35 degrees Celsius. Advantages for testing fastidious bacteria and identifying MBC endpoint. So, disadvantage, this is impractical when used to test several antimicrobial agents or isolate. Kaya mas ginagamit yung ito, broth microdilution. So, the same medium MH broth. Okay, yung volume naman, 0.05 to 1 ml. Ano ulit sa macrodilution? Greater or equal than 1 ml. So, standard inoculum size, the same. Okay, but for anaerobic bacteria, you will use 1 times 10 raised to 6 CFU per ml. So, incubation time, 16 to 24 hours, 35 degrees Celsius. Okay, and in anaerobic bacteria or anaerobes, 48 hours at 35. So, advantages for different antimicrobials. So, you can use 10 to 15 antimicrobials at the same time against a single isolate. Okay, disadvantage, inability to produce penicillin MIC that is consistently inside the resistant range for staphylococci. So, yeah, dapat dumiretso na lang pala tayo dito para mas clear. Kasi, being a medtech, dapat alam yung mga laboratory methods. So, first, kailangan mo mag-prepare ng McFarland. Okay, when we say McFarland, that's the standard turbidity or standard concentration or optical density ng medium and the bacteria. Okay, so yung unang picture dyan, so here sa medium, this is your Mueller-Hinton broth. Okay, maglalagay ka ng colony ng bacteria. Okay, kapag microdilution, ano ulit? Yung colony size, 5 times 10 raised to 5 CFU per ml. Tama? So, sa plate, kuha ka ng colony, lagay mo dyan sa MH broth, then mix. Then kailangan ma-meet niya yung turbidity. So, this is the standard turbidity or the McFarland standard. Ito lang yung ginagamit. Okay? 1, 2, 3, 4, hindi yan. So, ito dapat mag-match. Sa in the lab, may basis na doon na prepared. 8.5 McFarland. So, ang gagawin mo na lang, your MH broth with the bacteria and your McFarland, i-compare mo na lang. So, kapag hindi ka sure, you can always use your spectrophotometry. Okay? Ang kanyang optical density is 0.5 dapat. Okay? Which is equal to 1 times 10 raised to 8 CFU per ml. Now, ang gagawin mo, so, then you will set aside your stock solution. Okay? Wait lang nga. Medyo nangkatin ng ilong ko. Okay, set aside your stock solution and you will serial dilute your antibiotic. So from 64 micrograms per ml, okay, kukuha ka ng mga 1 ml or 2 ml, then ililipat mo. Napansin niyo meron na siyang mayroon na siyang distilled water. So ililipat, you will serially dilute from 64 to 32 to 1 to 16 to 8 to 4 to 2. And with decreasing concentration of antibiotics na to. So dito, syempre, pinakamaliit dito. Then, ililipat mo yan, no? Dilutions of drugs are made by preparing the serial dilution, then transfer mo yan sa 96 well plate. So, your 96 well plate, yan itong itsura niya. Okay, honestly, okay, di pa ako nakatry gumamit nito, but to show you, para hindi na tayo, may ganito pala. Pero may ganitong SMU ata. Hindi lang tinuro sa amin. So, this is your 96 well plate. Okay, maglilipat ka dyan. Okay, so ito yung 32, 16, 18, and 1. And of course, bago ka mag-proceed, meron ka ring growth control and sterile control. Okay. Wait lang, balik natin. So from here, ilalagay mo na yung 0.5 McFarland, yung stock solution mo, which is your Mueller-Hinton plus your bacteria. So ilalagay mo siya dito, 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 at dito. With the same amount. Okay, hindi pwedeng mas maraming ilalagay mo dito compared dito. Okay, the same amount. So ganyan ang itsura niya. Kunwari, nailagay mo na. So naganda na yung kulay. Ngayon, bago mo i-incubate, dapat meron ka ding growth control and sterile control. Siyempre, pag may growth control, walang antibiotic. Kapag sterile control, walang bacteria. So, dapat, pagkalabas mo ng incubator sa growth control mo, siyempre, dapat may growth. Sa sterile control, dapat walang growth. So, natatandaan nyo pa ba yung bacterial growth curve? So, lag, log, exponential, and decline. Okay. So, in this case, kunwari sa 32, okay, wala siyang growth. So, dito, walang growth. Na-inhibit niya lahat ng bacteria, ibig sabihin, clear. Hindi siya turbid. So, the same with 16, wala ding growth. Sa 8, wala ding growth. Sa 4, walang growth. Pero sa mababang concentration ng drug, which is 2 micrograms and 1 microgram per ml, may growth na. Okay? So from the result, so saan sa tingin niyo yung minimum, yung MIC, minimum inhibitory concentration? Saan yung pinakamababang concentration that can still inhibit the growth of your bacteria? Saan? Anyone? Sige na, para
4,
Sir,
sir.
4, sir.
4. Ayan, tama. 4. So, ganun lang yun. Madali. Dali lang. Huwag niyo kasi isipin mahirap. So, ganito yung itsura niya. So, dito, saan dito yung MIC?
8 po.
8. At dito naman, hindi siya ata clear. So, 8, dito, dito, itong tube na to, di ba? 1.6 micrograms. Ganun lang yung broth dilution. It determines the MIC. You will first prepare your stock solution, MH broth plus bacteria, then set aside, then mag-serial dilute ka ng antibiotic mo. Okay? Then, ililipat mo sa 96 plate. Kung wala kang plate, you can always use mga test tubes. But this is macro dilution. Okay? Then, you incubate. Okay, syempre may label tong mga to hindi pwedeng walang label. Sabi ko nga last time, ang buhay ng medtech, labeling. Okay? And you incubate, then tignan mo yung growth, kinabukasan, kung ganito, kung walang, kunwari ito yung growth na, wala siyang growth dito sa 8. So your MIC is 8. Okay, nakuha naman natin lahat. So properly preparing inocular are vital in the accuracy of the results of the antimicrobial testing. So during inoculations, agar, all bacteria must be carefully deposited directly into the antimicrobial solution. Next is your agar dilution method. So, kung may broth dilution, meron ding agar dilution. Ito yung reference method. Sa mga textbooks nyo, kapag reference method nakalagay, tapos may question, tapos may nakita kayong question na gold standard. So, gold standard or reference method is same. Okay, so the gold standard for testing anaerobes and Neisseria gonorrhoeae is your agar dilution method. This is frequently used in research studies because of its accuracy and reproducibility. So medium for anaerobes is BLSB or your Brucella leak-shaped blood or Wadsworth method. Using Wadsworth method, susceptibility medium, the same MHA. Standard inoculum, 1 × 10⁻⁴ CFU per ml. Incubation, 35 at 48. Okay, agar dilution plate shelf life is 1 week. Okay, advantageous for testing 1 or more bacterial isolates and for fastidious bacteria. Okay, so agar dilution, ginagamit to if single antibiotic ang tinetest against a number of bacteria. Okay? So this is the method, no? You have first to prepare your stock solution. So you have your antibiotic. Most of your antibiotics are available in 600 milligrams per ml. 600 milligrams. So usually available siya sa ganyang milligrams. So you will dilute the 600 milligrams to 6 ml distilled water to make a stock solution having a concentration of 100 milligrams per ml. So from here, your stock solution, okay, kukuha ka ng 1 ml and transfer to another tube with 9 ml distilled water. So ngayon, nag-transfer ka dito, magkakaroon ka ng 1 ml, dati 9 ml to, di ba? Magkakaroon ka ng 10 ml, 10 milligrams per ml concentration of your antibiotic. Okay, from 10 milligrams ml solution, aliquots, gagawa ka ng aliquots. Okay, from here, you will make an aliquot at 1 ml. Then after preparing yung stock solution, you will set aside your aliquots. Then you will prepare antibiotic dilution. Okay, you will perform antibiotic dilution. So let's take your aliquot for example. So 10 milligrams, ang concentration niya is 10 milligrams per ml. So ita-transfer mo yung 1 ml. Okay? You will transfer 1 ml into the tube having 9 ml of distilled water. So, di ba 1 ml to? Ita-transfer mo sa 9 ml distilled water. Then you will have 1 milligrams per ml dilution. Okay? Tama? Sige, 1 ml plus 9 ml, 10 ml with the concentration of 1 milligrams per ml. Okay, now, from 1 milligrams per ml concentration, okay, you will take, let's take for example, 800 micrometer, okay, and ita-transfer mo sa 19.2 ml distilled water. So, 800, kukuha ka ng 800 microliter using your, syempre, micropipette na yan. Lilipat mo siya sa 19.2 ml distilled water. Okay, this will give you 20 ml. Okay, with a concentration of 4 micrograms per ml. Then, you will now perform serial dilution. Okay? So, di ba 4 micrograms? Here, 4 micrograms, you will dilute. Kuha ka ulit ng 800 microliter. Lilipat mo. Okay. Ililipat mo, yung concentration mababawasan, lilipat ka ulit, mababawasan, maglilipat ka ulit, mababawasan. Performing your serial dilution. Okay? Then, you will use, ayun, Mullen-Hinton agar. Ito, liquid form pa lang to. Yung serial, okay, hindi lang iisa, okay. Kung ilan ito, kunwari, meron kang 4 micrograms, 2, 1, and 0.5. kung ilan yan, dapat ganun din ang bilang ng ganito, Mueller-Hinton-Agar. Nakikita nyo naman yung arrow, di ba? Okay. Kung 4 micrograms to, ang ihahalo mo dapat sa kanya is yung may 4 micrograms per ml concentration of antibiotic solution. So this one. Paano pag ito ang meron? Dapat ang concentration naman nito is 2 micrograms per ml. So you will mix and you will put it in the plate. Then you will cool para maging plate, para maging agar. Okay. Then, you will set aside naman para mag-solidify yung agar. And after you prepare your agar, you prepare your inoculum. So, iba ito ito, hindi ito. Hindi ito. Okay? This is from a separate plate kasi kunwari ang granome is stuff out. So, kuha ka ng isolate kasi kailangan mong mag-prepare ng inoculum. So, kuha ka ng isolate, you will pick a colony, okay, and add into the distilled water to make a suspension. So, dito, kailangan mo siyang i-further dilute. For the sake of our discussion, ang commonly used ginagamit naman is yung McFarland. Okay? So, i-compare mo sa comparator chart. Okay, dapat ma-reach niya or kung wala kayong comparator chart, you will use spectrophotometry with 0.5 optical density. Okay? Pagod no? Then, inoculate. Mag-inoculate ka ngayon from your inoculum. Tama? Ng 1.2 per microliter onto the agar surface. Then, you incubate the procedure for different concentrations. Then, i-incubate mo na. Okay, ang tatandaan nyo lang naman sa agar dilution, ang gulo no sa umpisa, pero yeah, I think hindi nyo naman na to kailangan. I think. Kasi in the laboratory, you will not performing agar dilution. Sa mismong work, I don't know lang sa klase nyo kung ipe-perform nyo. Sa laboratory, ang ginagawang common nilang, sabi ko nga, is yung Kirby-Bauer test. So just to give you the idea, may agar dilution method tayo wherein yung agar plate is may concentration siya ng antimicrobial. Ngayon, doon ka maglalagay ng isolate. Okay, ang tandaan nyo lang, basta ang agar dilution, this involves incorporation of different concentrations ng antimicrobial substance into an agar medium. So, yung agar medium mo may antimicrobial agent. Okay, and followed by the application of standardized number of cells from your inoculum. Okay, which in this example, gumamit tayo ng 0.5 McFarland. Okay, for interpretation, actually, nahirapan ako maghanap. So, I want to make this as your assignment. May nahanap na ako, pero wala lang. Gusto ko rin malaman nyo kung paano i-identify yung agar dilution. Hindi nyo ito isasubmit sa LMS, but can be a graded recitation on... Hindi, submit nyo na lang pala sa LMS kasi parang ang unfair pag isa lang yung nag-recite tapos lahat naman gumawa. So, assignment will be posted on the LMS later. So, interpretation of agar dilution method. Okay. Ito yung pinakatumbok ng discussion natin, the disk diffusion method. So, this is also known as the Kirby-Bauer test, limited to aerobic or facultatively anaerobic bacteria. Qualitative method that provides the greatest flexibility and cost-effectiveness that allows the laboratory to test 12 antimicrobial agents on 150 mm MHA plate. Meron din tayong 70 mm MHA plate, kaso hindi siya 12 microbial agents. 6 microbial agents lang siya. So the principle, syempre, the larger the zone of inhibition, the lower the MIC. So the zone of inhibition is inversely related to MIC. So remember the principle. Okay, habang lumalaki yung inhibition, bumababa yung MIC. Okay, standard medium, MHA. So yung zone of inhibition kasi, parang pag malaki masyadong effect yung inhibition, super effective yung antibiotic drug. Therefore, mas kailangan mo ng mas maliit na concentration niya kasi super effective na nga siya. Kaya mas lower yung MIC. Parang ganun lang. Okay? So, standard medium, your MHA. Standard inoculum size, ayan, 1.5 times 8 times 10 raised to 8. So, dalawang principle. Okay, principle, antimicrobial discs are carefully placed. Okay, alisin yung principle dyan. So, antimicrobial discs are carefully placed on an agar that's previously inoculated with bacteria being tested. Alam nyo naman na kung paano i-strict ang MHA, di ba? I think we've tackled about this in culture and culture medias. Tama? The disadvantage, not recommended for testing slow-growing organisms like mycobacteria and anaerobes. Diba? Baka nag-diffuse na lahat ng antibiotic nyo, hindi pa rin nag-grow yung mycobacteria. Yung mycobacteria or yung mga slow growers natin. So, your turbidity standard, okay, yung 0.5 McFarland natin. So, yung parang ginamit din nating turbidity sa agar dilution method. Okay. So, your turbidity standard is your barium sulfate suspension composed of 99.5 ml of 1% H2SO4 and 0.5 ml of 1.175 of barium chloride. So, this is equivalent to 1.5 times 10 raised to 8 bacterial cells or colony forming unit per ml. So, pure cultures are grown on or are directly prepared on an agar plate to match the turbidity of the McFarland standard. So, always check the turbidity pag nagmamatch siya sa McFarland standard na meron ang laboratory nyo. Kung mayaman ang lab or kung marami kang time, pwede ka namang gumamit ng spectrophotometer. Okay? Ipapasok mo lang sa spectrophotometer, then dapat ang optical density is 0.5. Siguro lahat naman alam na ang spectro, di ba? Lahat naman kayo may CC lab. Tama? Okay. So the preparation of pure culture for disc diffusion, okay, as we have discussed then. So 45 inoculum pure culture inoculated in triticase soy broth and incubated at 35 degrees Celsius for 18 to 24 hours. So your triticase soy broth is yellow kasi siya. Tapos wala akong makitang example na McFarland na yellow. So yung dito, kunwari yellow na lang tapos nag-match sila. So bacterial suspension and McFarland solution are compared by matching the turbidity of the tube against a dark background. Okay, if the bacterial suspension initially match the standard, okay, pwede mong i-dilute or pwede mong dagdagan ng bacteria. I-dilute mo kung mas turbid siya or lalagyan mo ng another colony of bacteria kung masyado naman siyang diluted just to match yung McFarland standard natin. Okay, during staging and placement of disc, alam niyo na ito lahat. So here, and I've mentioned before that each laboratory have their own protocols. So, yeah, ang ginagawa sa amin sa CVMC noon, ulitin ko na lang, baka hindi na. Meron pa ba yung, ayun, meron pa yung akip. Okay. Ulitin ko lang. So, let's see, this is your MHA. Yeah, your Hinton agar. Dapat alam nyo rin humawak ng plate. Okay? So, sa CVMC, they have their protocol na kunwari ito yung kunwari ito yung swab. Okay. They have their protocol na ang gagawin, maglalagay ka muna daw ng cross. Then you will make a lawn. Isuswab mo siya, then iikot mo naman sa kabila, then isuswab. Okay, then idadaan mo yung swab dito para lahat ng area is na-cover. Ngayon, pag hindi ka pa kontento, pwede mong gawin ulit, basta symmetrical lang. Kung ano yung ginawa mo sa isa, gawin mo sa opposite. Okay, so dito sa example natin, so yung A, nag-streak siya pa ganito, then inikot, hindi, kalahati lang then inikot niya pabaliktad, nag-streak siya naman. Then, inikot niya, so ito yung primary streaking, yung pag ganito, then nag-secondary streaking, then inikot niya ulit. Then, pwede mong idaan dito yung swab para lahat ng ears na cover. Pasado na ba to? Kung para sa akin, gusto ko pang lagyan ng isa dito. So, ganito na pasay at isa din dito. para symmetrical. Okay, ganun lang yan. So, surface of the medium should be allowed to dry for 3 to 5 minutes, no longer than 15 minutes. So, in preparation of your disk diffusion method, within 15 minutes lang. Okay, disk must be positioned no closer than 24 millimeter from disk center to center. When we say center to center, from the center of the disk, kunwari, ito yung disk. Okay, kunwari lang, ito yung antimicrobial disk. This is the center. Then another disk. This is the center. So, ang center to center is from here to here. Ang dapat layo ay 24 millimeter. So, discs are pressed firmly to ensure contact with agar. So, within 15 minutes of disc placement, plates are inverted and incubated at 35 degrees Celsius for 16 to 18 hours to a maximum of 24 hours. So, this is the process. So, you have your isolate. Okay? Let's say ito na match na yung McFarland. Okay, then kuha ka ng swab, isawsaw mo dyan, then ang ginagawa kasi sa lab para hindi siya basa-basa, i-press mo siya sa gilid ng tube para maalis yung ibang moist. Then you will do the staking. Okay, again, different protocols. Pero not compromising the quality of your test. Okay, then ang ginawa niya, ayun, sinab niya rin sa gilid to ensure. And nag-place na siya ng disk. Okay, nag-place na siya, then press niya firmly para hindi maalis-alis. Okay, remember, ang disk positioning is no closer than 24 millimeter from disk center to center. Okay, pag center to center, as mentioned kanina, at yung distance naman from the edge to edge, okay, is 10 to 15 millimeter. So, edge to edge, 10 to 15 millimeter. Pag center to center, 24 millimeter. Okay, and yan, innovation. Hindi ko pa na-try ito, wala rin ganito sa CVMC, pero meron nang gumagamit ng ganito. So, I think ang kaya nito is 6 antimicrobials. So, ito yung mga antibiotic disk natin. I-insert mo lang sa dyan. Then, ang gagawin mo, ipipisilin mo. Actually, may kwan ito, may ganyan kahaba dyan. Then, pipisilin mo, then, bababan na lahat ng antibiotic. And then, you will incubate. Huwag mong kalimutan i-invert. Ang purpose ng inversion, pag in-incubate mo yan ng hindi naka-invert, mag-moist dito. Ang mangyayari sa moisture, mababasa yung antibiotic disk, wala na, hindi na quality yung result. So, kaya kailangan mo ang i-press para firm na nakadikit yung antibiotic disk mo para pag in-invert mo, hindi siya mahuhulog. Okay? Dito, I don't know yung prinsipyo nito kung aabot ba siya mismo sa agar o mapipress na or mahuhulog na lang basta-basta. Pero, of course, yung mga gumagawa ng supply, ine-ensure naman nila yon. So, siguro may pressure din na onte para ma-press yung antibiotic disk sa surface ng agar. And then you will incubate. Ang kalalabasan, ganito. So, ang choose ni clinician dito, hindi na siya magdalawang isip na piliin to. I-reject niya na to agad. Okay, then you will measure the zone of inhibition using a ruler or vernier caliper. Hindi ko nalagyan kung paano mag-measure. Pero, sige, next meeting, gagawa ako ng slide on how to read vernier, the millimeter, the zone of inhibition during vernier caliper. Okay? Or baka sa lab nyo, naituro na, so better. Sabihin niya na ako kapag naituro na para hindi na ako uli. Okay, pwede kang gumamit ng ruler or pwede kang, and better naman pag gumamit ka ng caliper. So, saan ka nagme-measure? Hindi sa baba, hindi sa puwet. So, syempre, ang gagawin mo, aalisin mo na, then pupunasan mo yung moisture ng takip. Kunwari ito yung takip, aalisin mo, pupunasan mo to, then babalik mo. Then you will measure from the top. Kung expert ka na, kahit nakabukas na yan, may measure mo na lang. Yung head namin sa CVMC noon ng backtape, ginaganyan-ganyan niya lang yung caliper. Ang bilis niya mag-measure. Ang galing. Okay. So reading of plate and interpretation of results. So law of growth must be confluent or almost confluent. Okay, lahat dapat ng areas makovera na dapat ng areas maganda yung growth. O, tignan nyo to, maganda yung growth lahat, di ba? Even. Okay. If growth is not confluent, test should be repeated. What if kapag dito wala siyang hindi siya na istrikan ng maayos. Error na yun, error na yung pagbabasa. Or let's say dito bandang hindi na istrikan ng maayos. Siyempre may error na sa pagbabasa dito, dito at dito na antibiotics. Okay? So galing-galingan lang natin sa pag-streak. Yun lang naman ang solusyon. So diameter of each inhibition zone is measured using a caliper or vernier caliper or ruler. Plates are measured 2 to 3 inches above a black non-reflective surface and zones are measured from back of the side or plate. Sabi ko nga, pag expert ka na, kahit nakabukas na yung plate mo, ganyan-ganyan mo na lang. Okay? And also, you can measure in front or from the back. Okay? Ang exceptions lang dito, may may MHA kasi na may blood. Example, this one. You can't measure it from the back. Diba? Kasi hindi mo na makikita yung growth. So, you will measure it sa harap. Okay? Pero pag MHA, you can measure from back or from front kung saan ka sanay. Okay? Basta hindi nako-compromise yung quality. Basta tandaan lang natin yun. In every method na gagawin mo, okay, make sure that the quality of your test is not compromised. Okay. So transmitted light rather than refracted light will improve the accuracy of test with penicillinase-resistant penicillins. For media that contains blood, the plate is examined with the lid removed. Okay? So, parang ito. Lid removed. Measuring. And zone measurement is compared with interpretative tables of CLSI. Okay, na-browse nyo ba yung sinend kong CLSI? So, maraming samples doon and the results are interpreted as susceptible, intermediate, or resistant. So, example, ganito. Let's take, for example, your amikacin. Ang resistant niya, less than 14. Ang intermediate, 15 to 16. Ang sensitive is 17. Okay. 14 and 17. Okay, example, this is your amikacin drug. Okay. Ang measurement dito is less than 14. So, nasa 12 lang. So, is this resistant or susceptible? So, resistant. Tama? Balikan nyo natin. Mali. Pag greater than 14, I mean, resistant. Hindi ko na pagsama sa slide. Okay, pag greater than 14, yung zone of inhibition dito, this is resistant. But, kapag, tama pala, ano ba yan? Anyways, greater, less than to, di ba? Greater. Okay. Let's take, for example, this is amikacin and yung growth dito is less than 14. So, less than 14 lang to. Hanggang dito lang, 14. So, ibig sabihin, yung bacteria is resistant to this. Ngayon, pag 17, mga dito na siguro, okay, 14, 17, greater than 17, a measurement using your caliper or ruler, this is interpreted as susceptible. Okay, let's take another example kasi medyo naguluhan ako din. Resistant, less than 15, greater than 21. Okay, kunwari ito, less than 15. Less than 15. So, you will measure from the edge to here, less than 15. Then, this is pag less than 15, resistant. Tama? Kapag greater than 21, yung growth, yung zone of inhibition, then this is susceptible. So, simple lang naman. Ang kailangan, ang susi lang dyan, dapat familiar tayo sa mga ito. Sa mga ito. Okay, familiar tayo dyan, familiar tayo dito. Dito kahit wag na. Okay, we just need to be familiar with this and this is just an example. You don't need to memorize itong mga to. no? And hindi ko na rin ipapamemorize kung ano yung nasa CLSI kasi hindi siya worth it. Okay? Sa laboratory naman, may guide kayo eh. Tsaka may CLSI doon na libro. Okay? May manual, so you don't need to memorize. You just have to know the principle kung paano mag-measure, kung ano ina-measure, kung saan ina-measure, at kung ilan yung dapat i-measure. Okay, reading of plates and interpretation of results. When we say susceptible, okay, the patient's infecting organism should respond to the therapy. So the microbe or the organism is susceptible to this certain antibiotic. Okay? Kapag intermediate, falls into the range of susceptibility in which MIC approaches or exceeds. Kapag resistant, walang zone of inhibition. Okay, ang tandaan lang, in contrast to your susceptible, yung intermediate is less clinical, okay? Less clinical response siya compared with susceptible strains. Kapag susceptible, syempre, yung respond is better. Okay? So, effective. Okay, control plates should be read prior to the interpretation of results of patient isolates to determine whether the test was performed correctly. So, causes of false resistance, so heavy inocula or undiluted specimen. So, masyadong marami kang inilagay, masyadong napadami yung in-strict mo. So, pwedeng hindi mo siya, ano yun, pagkukuha ka kasi sa tube, idadaan mo siya sa gilid. Yung puspusin yung cotton para maalis yung ibang tubig or yung ibang liquid doon sa trypticase. Pero ang ginawa mo, pinasok mo na lang, hindi mo na pinispis sa loob, then in-inoculate mo agad. So, ang mangyayari, napadami yung paglagay mo ng inocula, napadami yung bacteria. Okay? Ngayon, ang mangyayari, yung antibiotic, ma-overcome siya kasi sobrang dami ng bacteria na nailagay mo. Okay? Pwede ring mali yung pag-follow mo ng standard McFarland. No? Undiluted siya masyado or masyado siyang dilute. Okay? Hindi na-follow yung standard. So, it's very imperative na yung comparator chart is na-follow, which is your 0.5 McFarland. Okay? Kaya dapat clear eyes pa rin tayo kapag nag-exam or kapag nagpo-produce ng test. Nagpo-produce ng AST test. Okay, late examinations of plate, ang tendency, okay, so pag sobrang tagal mong in-intubate yan, imbis na resistant, imbis na susceptible, nagiging resistance siya. So, false resistance. So, originally, susceptible siya, pero due to prolonged incubation, nagkaroon ng false resistance. Nagigets nyo? Na-imagine nyo ba? Okay. Pwede rin use of disc with inadequate drug concentration. Okay. Common cause nito, expired na yung drug. Or hindi siya na-store ng maayos. Or false resistance is caused by failure to immediately refrigerate the disc after use and use of wrong bacterial isolate. So notes, okay, high yield drug concentration decreases at increasing distances from the disc, as mentioned from your eagle effect. Diba? A wide zone surrounding a disc signifies a greater susceptibility of the organism to the antibiotics, and zone width is related to antibiotic concentration, diffusion rate, and solubility. Colonies appearing within a clear zone as a result of contamination or presence of mixed culture should not be ignored. Original colony should be tested. Actually, mahirap talagang ituro tong mga method kasi methods to eh. Ang hirap pag wala siyang application. So, mas magiging clear tong mga to sa laboratory nyo. Hopefully, tuturuan din kayo kung paano gumawa or mag-perform ng disc diffusion. Disc diffusion method, okay na. Okay. Zone size of swarming organisms like proteus should be ignored. Always ignore. Okay, when reading the era of inhibition. So routine susceptibility testing on beta hemolytic streptococcus is generally unnecessary. Factors affecting the zone of inhibition can be the amount of inoculum or test organism. Pag masyadong makapal, false resistance. Pag masyadong manipis or masyadong makapal yung plate, Okay? Yung sa number 1 na pag masyadong madami yung nalagay mong inocula, false resistance. Pag masyadong makapal naman yung plate mo, ibig sabihin mas maraming nutrients or mas maraming supply na available sa bacteria. So, more nutrient to bacteria, syempre more growth. Therefore, yung zone of inhibition na dapat mapipigilan niya naman, pero sa sobrang dami, dahil napadami ka ng nutrients sa agar mo, nag-grow yung bacteria mo, magkakaroon ka tuloy ng smaller zone of inhibition. The same thing with too thin. Kapag too thin, sobrang nipis ng MHA mo, ibig sabihin kulang yung supply or kulang yung nutrient ng bacteria. So, ano ba ang mangyayari pag kulang ang nutrition ng bacteria? Mabilis siyang mag-decline. So, may hirapang tumubo. Therefore, the zone sizes will be larger. Yung inhibition is larger. Okay, nakuha natin. Ganun lang yon. Sobrang kapal, zone will be smaller. Sobrang nipis, zone will be larger. Okay, growth rate of test organisms. So 35 degrees Celsius is recommended for most bacteria. Lower temperature may result to larger zone of inhibition. Bakit? Remember that lower temperature is bacteriostatic. Okay, pag hindi niya na-meet yung incubation requirement niya, requirement temperature, may hirapang mag-grow si bacteria. Okay, increased carbon dioxide required for Neisseria meningitis and strep pneumoniae. So pH of medium. Okay, incubation of plates in carbon dioxide could result in decreased pH. So increased pH results in decreased activity of tetracycline drugs. Decreased pH results in diminished activity of aminoglycosides and erythromycin. The number of discs per plate, yung 150 mm plate can accommodate 12 discs. Now, I'm not sure kung 60 or 70 yung 6 discs. Pero, yeah, in your internship, magiging familiar ka sa lahat na to. So, this is your, let's say 70 mm disc. 1, 2, 3, 4, 5, 6, 7. And, kapag sobrang dami ng nilagay mo dito, magkakaroon ng overlapping of growth. Ito, overlapping of growth. Okay, so mahirap na siyang basahin. Pero in this case, pwede mo ba siyang mabasa naman? So from here, from this point to that point. Okay, from this point to that point. Okay, pag dito naman, kahit may overlapping from this point to this point or this point to this point. Wala kasi ako mahanap sa net ng error. So ito lang nakita ko na nag-overlap. So ito, pwede pa naman to. Pero as much as possible, dapat ang end ng plate natin after incubation ganito. Diba? Ang daling magbasa. So factors affecting the concentration of divalent cations, which is your calcium and magnesium. They can affect the testing of the aminoglycosides and tetracycline against Pseudomonas aeruginosa, and the elevated concentration of your calcium and magnesium may result in diminished activity of aminoglycosides. Next is the E-test. Actually, the same lang to sa antibiotic disc. So, should we read? Dilution test that is based on the definition of a continuous varying concentration of antimicrobial agent. Use of thin plastic strip that are impregnated with an antibiotic of decreasing concentration. Each strip is composed of single antibiotic with decreasing concentration. Okay, and the procedure. So, ang gagawin mo, okay, you will make a lawn first. Okay, alam niyo naman na kung paano gumawa ng lawn. At ito yung i-strip natin. So, ilalagay mo siya. So, let's take this for example, mas clear. So, i-test strip, parang kwan lang yan. Ay, wala pala yung arrow. So, this is the e-strip. E-strip, e-strip, e-strip. So, dito, yung pinakamataas na concentration of drug, then pababa, some very low. So, from 32, which is a very high concentration of drug, to 0.002, which is a very low concentration of drug. So, your e-test strip, para lang yan antibiotic disc, except that sa disc kasi, yung antibiotic concentration is homogeneous. When we say homogeneous, even. So, evenly distributed. In here, sa strip, on the other hand, as mentioned kanina, so mag-start siya from a very low concentration, which is from here, you have your 0.002. Okay, that works up to a very high concentration, 32. concentration gradient and is not circular but elliptical. Ayan, ellipse. So, ellipse, ellipse, ellipse. Hindi ko pa ito na-try. Honestly. Okay, an in vitro measurement of and wait lang, yung interpretation pa pala. Ito, yung intersection niya, yun yung measure, yung MIC mo. This is your MIC. So, your MIC is here. MIC. MIC. Ayan. Basta dito, yung point na nag-intersect sila dito is your MIC. Okay. An in vitro measurement of, next is your minimum bactericidal concentration test. In vitro measurement of the amount of antimicrobial agent required to kill bacteria. Standard inoculum, 5 times 10 raised to 5 CFU per ml. So this is the procedure. So in 0.01 ml aliquot of each clear MIC tube or well is subcultured to an agar medium, usually blood agar plate, and incubate overnight. So the volume spread per plate, 0.1 ml. Result, pag positive, 99.9% reduction in the CFU per ml as compared to the concentration in the original inocula, which is recorded as MBC. We will interpret. So if the number of the colonies on a subculture plate is less than 0.1% of initial inocula, indicating 99.99% reduction in CFU per ml, a bactericidal effect has been achieved. So just remember, ano bang definition ng MBC natin? Lowest concentration at which the antimicrobial disk will kill, di ba? So your minimum bactericidal concentration test, this, just remember na this determines the lower concentration at which antimicrobial agent will kill a particular organism. Paulit-ulit na tayo sa MIC at MBC. MBC and MIC. But it's better, di ba? One key of remembering is repetition. So your MBC test, your time kill assay, synergy test, mga test lang yan, but actually you won't be needing this, but it's very important that you have even the slightest idea lang. Okay, time kill assay, this involves the addition of bacterial isolate to an antibiotic concentration in a broth medium and measurement of bacterial killing rate over a specified time. So your time kill assay, okay, this is used to study, okay, the activity of antimicrobial agent against a bacterial strain and can determine yung bactericidal or bacteriostatic activity of an agent over time. Okay, over time. So how? You will test the bacteria inoculated into several broth tubes that contain various concentrations of antibiotic. You will incubate at 35 degrees Celsius, then small aliquots are diluted and plated for colony count. Bago ka naman mag-count, syempre, pag i-clinate mo na, i-incubate mo ulit. So, result, 3 or more log reduction in bacterial count in the test plate as compared with the control plate, which indicates an adequate bactericidal response. So, sir, kung hindi namin kailangan, ilalabas mo pa ba yan sa mga quizzes? Sure, ilalabas ko to sa mga quizzes. Pero ang tanong lang doon, okay, let's just limit it. Kunwari, this is interpreted as the number of colonies on a subcultured plate is less than 0.1% of the initial inocula, indicating 99.99% reduction in the CFU per ml. So your answer is minimum bactericidal concentration. And same here and here. Okay? So this synergy test, this determines the effectiveness of combined. So dalawang antimicrobials against a single bacterial isolate. This is performed using a broth dilution checkerboard method or time kill assay. Procedure. Okay, this is the procedure. So this A and B, these are filter papers. Okay, yung filter paper mo, na-soak siya sa agent A at or above MAC. So, kunwari ito yung filter paper, this is your tube. Iso-soak mo siya, aalisin mo siya, then ilalagay mo sa plate. Then, ganun din. Kunwari ito yung antimicrobial A and antimicrobial B. So, yung filter paper strip A, dito mo sinoak. Filter strip B, dito mo sinoak. Ngayon, you will place it in a plate dapat may synergy sila. Parang ganyan. Yung iba ang ginagawa cross or X. Okay, then this is the zone of inhibition. From here to here, okay, you will measure the zone of inhibition. Okay, that is your synergy test. Okay. Basta tandaan nyo na lang pag synergy. In vitro ba to? Yes, in vitro interaction kasi performed in lab of the in vitro reaction of antimicrobial combinations, okay, to determine whether the effect of the two, okay, in your example, antibiotic A, antibiotic B, of these two antimicrobials is greater than the sum of their individual activities. So, pinaghalong kapangyarihan ng filter paper disc A soaked in agent A and filter paper B soaked in agent B. Synergy. And your serum bactericidal test or your Schlichter test is similar to MIC and NBC test. It is inhibitory and bactericidal parameters that is being evaluated. This also determines the activity of one or more antimicrobial agents that are present in the serum against an organism that is obtained from one patient. Okay, final inoculum size 5 times 10 raised to 5 bacterial cells or CFU/ml per ml. Procedure, so an aliquot of each dilution is placed on the sheep's blood and then intubated. So result, the highest dilution that inhibits the visible growth is the serum static tier. Interpret the serum dilution which results in a 99% reduction in CFU per ml as compared with the original inoculum is the serum static titer. Okay, get familiarized with this lang naman. Synergy, time kill assay, minimum bactericidal test, e-test. Okay. Soon, no? Baka sa second or third meeting, i-discuss natin kung para kanino si e-test. Ano ang mas better gamitin sa ganitong bacteria, kung e-test ba or broth dilution. And we proceed to automation. Ang alam ko lang ng automation dito yung Vitec. Okay, meron kasi ito sa CVMC. This one, Vitec. Pero syempre, pag intern ka, hindi ka pwedeng gumamit. Mga stuff lang. So automation, they follow the principle of turbidimetric detection of bacterial growth in a broth medium by using photometer to examine the test wells. So you will interpret the growth and points of broth microdilution panels are detected using an automated reader device. So syempre, ang advantages naman most ng automations natin is shortened analysis for convenience. Ganon. So examples ng automated machines, in bacteriology section, you have your Phoenix system, Microscan, walkaway system, your Trek Sensititer, and Vitec 1 and 2 systems. Vitec 1 and Vitec 2 systems. So growth detection can also be performed through hydrolysis of fluorogenic growth substrate that is incorporated in a special medium. So we go to your examples. Your Phoenix system, okay, this uses a gravity-based inoculation technique, which I never know, and I never use Phoenix system. Okay, pero ang gamit sa Phoenix system and Microscan is this one. Yung 96 plate, may sarili silang plate. Okay, the microdilution tray. Then, i-incubate mo siya. Ang gagawin mo lang naman dyan, ilalagay mo yung, di ba, you have prepared na your inoculum, ilalagay mo dyan, your McFarland, okay, yung concentration of antibiotics, then ipapakain mo lang sa machine. Then, siya na yung bahala. Ano yun? So, like your Microscan walkaway system, inocula are manually introduced into the broth microdilution tray. So, ilalagay mo dito. Then, incubated. Pasok mo sa machine. Growth patterns are automatically read and interpreted. Si machine na ang bahala. So, bacterial growth could be detected using spectrophotometry and fluorometry. So, ano ang principle ng Microscan walkaway? Spectrophotometry and fluorometry. And your Vitec, Okay, ang kaiba naman dito, hindi siya microdilution tray. Ang ginagamit dito is ito. Plate. Card. Card. So, card siya. Ilalagay mo siya dyan. Mga cards to. Card. Then another card. Another card. Another card. Another card. Okay. Inocular are mechanically added to specified concentration of antibiotics. Placed in a miniaturized plastic closed card. Then, ipapakain mo na sa Bitec 2. Optical readings are made every 15 minutes to measure transmitted light through each well. So 64 wells are used. MIC and antimicrobial testing results can be identified using the advanced expert system in your Vitec systems. So, this is my last slide. Any questions, clarifications? May tanong, paki-on niya yung camera natin. Baka naman salita ako ng salita dito. Wala akong palang kausap. Yes, paki-on. At the-base ko dito yung attendance.
May quiz po ba after nito? If ever na meron, sir, okay lang next. Ang hirap i-absorb lahat.
Sige. Mahirap talaga i-absorb dahil sabi ko nga, lab methods mga yon. Classmates yung Marigold, kasama mo ba yung bang classmate mo? Yes. And nakalagin sila sa
Ano,
Gmeet?
sir?
Kasama mo
Yes,
yung ibang
sir.
classmate
Yes,
mo,
sir.
right?
Yes, sir.
Nakalagin naman sila sa Gmeet?
Yes, sir. Yes, sir. Yung ano, sir. Yung iba, sir, hindi kaya talaga ng ano dito, sir.
It's okay. Okay, pwede kayong
Kaya ano
parang
na lang sila dito sa laptop ko, sir.
better, di ba? Kung yung mga naki face-to-face, you can have your data. Kasi alam naman natin na yung Wi-Fi sa SMU
Okay, sir.
Google pa lang, ayaw na. Di ba? So, kung may face-to-face kayo, better. Okay, 50, okay na yun. 100, 1 week, pwede na yun. Okay, so, kung nasa school kayo, pwede na kayo hindi mag-log in sa Gmeet as long as, yun, parang yung ginawa nila Miss Marigold. As one. I think mas better pa nga pag ganun, di ba? Pero pandemic, hindi naman lahat have the opportunity para makalabas. So, okay, next week is your post-quiz. Let's say 15 items. And next week, 2 topics. Parang yung dati na. Okay, next week, release of grades. Okay, so, yeah. So, wala nang tanong. Thank you for joining your class and bye-bye. See you next week.
Thank you, sir. Bye, sir.
Thank
Bye, sir.
you.
Thank you po.